All right, so we're going to talk today about antibiotic resistance and whether we are headed into a post-antibiotic era. All right, that's what we care about. And I know right now it's a little hard to think about another potential disaster when we're in the middle of a pandemic, but I want you to really see that your evolution knowledge, that the knowledge that you've gained in this class can be used to think about policies and to think about ways that we can keep things from happening, right? And to improve human health and a bunch of other things. And so that's what we're going to work on today. So we're going to start with not really a knowledge check, but a quick check-in with a couple of questions. So go ahead and look at the questions on the left, and then we'll come back together. Okay, in this last module, our last of the semester, yay us for making it, we're going to focus on applying evolution to a problem in society. And so I'm going to introduce the problem, and then I'm going to talk about the origins of antibiotic resistance, how it spreads, and then we're going to speculate about how we can reduce the spread, and will reducing the spread actually help us with the problem. In the modern world, we kind of take antibiotics for granted. They're easy to get when we need them. But if we just go back a couple generations, if you go back to your grandparents' era, there were people dying from common infections like pneumonia, meningitis, and bacterial infections in wounds. In fact, 30% of all deaths were bacterially related in the prebiotic America, and it really wasn't that long ago. And the discovery of antibiotics was so exciting. So in this quote by Charles Fletcher, who was basically a student who had just come out of med school and was participating in early penicillin trials, he says, "It's very difficult to convey the excitement of actually witnessing the amazing power of penicillin over infections, for which there had previously been no effective treatment. I did glimpse, kind of in that moment, the disappearance of the chamber of horrors, which seems to be the best way to describe those old septic wards." So septic wards are where they took people when they had these bacterial infections. And could see that we would never again have to fear various different bacteria again, right? So antibiotics were revolutionary. Even now, we use antibiotics for things that you might not immediately think of. So obviously, we use them for the treatment of common infections, right? That's what we think of maybe most of the time. But they're also involved in a bunch of advanced medical treatments, things like chemotherapy, organ transplants. When you put in medical devices or prostheses, they're involved in neonatal care and invasive surgeries. If they stop working, all of these things become so much more risky. And right now, we think they're going to stop working, possibly in the next 10 years. So the World Health Organization has said antibiotic resistance is one of the biggest threats to global health, food security, and development today. It has been dubbed the end of medicine, and it may be the grand challenge of our age to try to keep antibiotics from going away, from not working anymore. And what's crazy is that people aren't really talking about this. Now, obviously, right now in this moment with the pandemic, I don't expect anyone to be talking about this. We're focused on COVID-19 and how we can deal with it, right? But think about before. How much were we talking about antibiotic resistance relative to, say, climate change or things like that? And I'm not saying one is more important than the other, but like this is going to impact millions and millions and millions of people. And how much were we hearing about it? So the first thing we need to do is define the problem. What is antibiotic resistance? Antibiotic resistance is when bacteria adapt genetically to the presence of antibiotics by acquiring various resistance mechanisms that keep the antibiotics from actually working anymore, from inhibiting the bacteria. So this means that resistant bacteria can still grow and reproduce in the presence of antibiotics, so totally nullifying, the benefits of antibiotics. That is the problem, and that is what we're trying to avoid. So to understand the problem, the first question we have to ask is, why is antibiotic resistance evolving in bacteria? And so I'm going to show you two graphs, and I want you to draw conclusions from these graphs related to that question. And so both of these graphs are going to show you on the x-axis use of a particular antibiotic. So penicillin here and vancomycin, I don't know how to pronounce that here, but two antibiotics. And that we're using them in different contexts. So this first one is being used within ICUs, so in hospitals and intensive care units. And then the other graph on penicillin is looking at a national level, so looking across 19 different countries in Europe. All right, so I'm going to pop the figures up for you to see them, and then a question is going to come up for you to interpret them. So what I hope you were able to see in both of these graphs is that as penicillin or as antibiotic use increases, so does resistance. As use increases, so does resistance. Right? And you can see it in both these figures. Evolutionary biologists would look at those graphs and be like, duh, of course it does, right? And so now I want you to tell me why. Why is that such an obvious thing, obvious relationship, exactly what we would expect? So go ahead and answer the question that pops up now. So scientists, when they see relationships like that as the use increases, so does resistance to it, think that's pretty obvious. And it's a pretty obvious conclusion of natural selection, right? So if we look at what's going on here, we have bacteria differ from each other in their resistance to an antibiotic. All right, postulate 1. Postulate 2 is these differences are genetic. So the differences in resistance are due to genes, not environment. It's a genetic thing. So if they're genetic, they're passed on from parent to offspring. And then antibiotic resistance causes some individuals to survive better than others in the presence of antibiotics. And those successful individuals aren't lucky. They succeed because they're resistant to the antibiotics. Right? So just those 4 postulates, which you already know, can explain why, as we use more antibiotics, and the bacteria are more likely to become resistant to them. Antibiotics exert a selective pressure on individuals that produces a variation in fitness based on whether the individual is able to tolerate and grow and survive in the presence of that antibiotic. It's just a selective agent. That's why scientists were like, of course this is happening. All right, so that was the problem, antibiotic resistance, evolving in the face of our use of antibiotics, but we still need antibiotics. So we need to further understand the problem so we can come up with some solutions. So now let's talk about where antibiotics come from and where the resistance might come from. So this is something I did not know. We do not make most of the antibiotics. We don't synthesize them out of nothing, right? We steal them from existing sources in nature. So antibiotics are a compound that inhibit or kill bacteria by some sort of interaction with some target in the bacterial cell. And most of them come from soil-dwelling bacteria and fungi. So things out in nature. So why would bacteria make antibiotics? That doesn't make a lot of sense, right? Well, they think it might help them inhibit competitors. So if you produce antibiotics, right, something that slows down the growth and survival of competitors, and you send it out into the environment, you could up your fitness. It could also be used in cell-to-cell signaling. All right, so these antibiotics exist in nature. And then we find them and we produce them on a large scale to use for us. Why does that origin matter? That it's found in nature? Well, many bacteria then have already had a long history of exposure to antibiotics. Even before we started using them, they encountered them in the environment, which means they've been under selection potentially for a long time, which has allowed them to maybe already develop resistance in some members of the population before humans even started using the antibiotics. So these interactions have been happening a long time before humans even started using it. So let's talk briefly now about where the resistance comes from. I want you to go through a couple questions now and think through, where does antibiotic resistance come from in a population? All right, let's debrief that on the next slide. So I hope you came up with the idea of mutation and migration. So mutation, right, is just a change in DNA. And for it to lead to antibiotic resistance, somehow that change in the DNA, which leads to a protein sequence change, confers some sort of resistance to antibiotics. But what do we know about mutations? We know that most mutations are random, and most end up either being deleterious or neutral. What's the chance that a mutation is going to lead to antibiotic resistance? It's incredibly small. That makes this process slow. What else slows this process down is what we talked about in genetic drift, where genetic drift eliminates rare alleles, right? So remember that your chance of spreading to fixation is as an allele is equal to 1 divided by, is equal to how frequent you are in the population, right? So if you're very, very rare, if you've appeared in one individual, you are very likely to be lost. That also slows down mutation's ability to be the origin of antibiotic resistance. So it happens, but it's slow, which is why it's so important to understand the problem, to know that antibiotic resistance or antibiotics have existed before humans. That has given time for some bacteria to develop resistance, right? It's been in soil, the soil bacteria has been producing it, and other bacteria have been reacting to it for thousands to millions of years. So mutation is our slow method. The other method is migration. So again, there's vast pools of resistance genes in many species of bacteria. And many of these could be co-opted to provide resistance to antibiotics used against humans and animal pathogens. So existing resistance genes in some populations could be useful to bacteria who are trying to resist antibiotics in humans. And that is a faster process because those genes already exist and they just have to be spread. We don't have to wait for the random chance of a mutation to produce one. It already exists in a population, it just has to move into yours. And that becomes much more likely in bacteria because of horizontal gene transfer. You can get genes transferring between individuals who are not related to each other. And so migration is a much more powerful force in bacteria because you're not just waiting for a migrant to breed with an individual in that population. Pieces of their DNA can migrate into an existing individual. All right, so that just even makes this faster. So when we're talking about speed, we're really concerned about migration. Mutation is a much slower process, but again, because these antibiotic resistance has been around so long, antibiotics have been around so long in soil bacteria, the resistance could have evolved a long time ago, and now we've created a selective pressure to cause it to spread. So let's talk about the spread. Migration isn't just happening in hospitals, right? So when you're in a hospital, the first thing I was like, well, when is someone in a hospital exposed to dirt bacteria so that genes can be exchanged with the bacteria in their bodies? But that's the thing. Antibiotics resistance is not just popping up in hospitals. And where a lot of it is coming from is transferring from agricultural animals to us. So we use millions of kilograms of antibiotics in our livestock, and we use them to promote growth and improve the conversion of food to growth. Because if your immune system is having to act to protect you, you're using energy in your immune system that you can't dedicate to growth. But if you can keep animals from ever getting sick, then all that energy can go into getting bigger and producing more meat, right? Which is desirable if you're farming animals. And I think what's really interesting to compare is the amount of antibiotics used in the agricultural industry versus in medicine. It's over 10 million kilograms more antibiotics are being used annually in the agricultural context. So this is a major source of selection pressure on antibiotics. They're migrating from animals, which we consume or we come into direct contact with, and then we get those bacteria and they mix with our bacteria. And there's evidence for this transfer. So studies have been done showing that bacteria in livestock are passing directly to farm workers who attend to the animals, and also that through food prep we can pass that bacteria. So if you eat raw meat, I love sushi, but this is one of the ways that you can pass bacteria. Or if you're just not cooking it sufficiently. My mom likes her steak bloody. That is a way to pass the bacteria that may have antibiotic resistance because it was facing antibiotics in all those organisms. So let's review how, again, antibiotic resistance might spread. So for a mutation, you need to have a mutation arise granting that antibiotic resistance, and then strong selection favoring that resistance so that it moves beyond one individual to become very common in the population. For migration, you have to have a migrant who already has the resistance mechanism. That resistance mechanism must have a genetic basis, And then there has to be opportunity for transfer between that migrant and an individual in the current population. And that transfer must mean that the individual who receives it becomes resistant. And then again, that's still just maybe 1 or 2 individuals in the population. So for it to spread and become common, you need strong enough selection to favor that resistant phenotype. So keeping those things in mind, think about this next question. So you may have come up with quite a few different things that are possible to influence the answer to this question. So I'm just going to jot down a couple. So host density. So we're seeing that as a problem now too, right? When cities are more hard hit by COVID than rural communities, right? So host density enhances the spread, and that's important for migration. Patterns of host travel. Again, this is about migration. Patterns of host travel, and we see this with COVID, the more you move between populations, the more likely that the resistance is going to get to move between populations. So if the host is traveling a lot and far distances, so is the resistance. Hygiene, right? Depending on how much people wash their hands or how good they are with food prep, you can either kill the bacteria or spread the bacteria. And then there is host immunity. We can also think about things on the bacteria side, such as how easy is it to transmit the resistance? How easy is it for another bacteria to pick it up, right? So there are a lot of things you could have written here about. These are just a couple. The other thing that we can think about when we're thinking about what can cause the spread is the strength and the context in which selection is experienced. So the stronger the selection at any moment, right, the stronger the antibiotics, the more they cause differences in fitness, the stronger the selection for antibiotic resistance. But also the more consistently you experience that selection pressure also can lead to more antibiotic resistance. So consistency in time as well as acuteness at one particular time. So the maintenance of antibiotic resistance in a population at large over time depends on resistance being at least neutral, where being resistant to antibiotics doesn't have selection against it, or ideally it's advantageous over time. And so the question is, do these conditions exist that would consistently provide a fitness advantage to antibiotic resistance? Because really, when we think about antibiotics, I think about, I got a cold, right? And they give me antibiotics because they think I got a bacterial infection with it, right? I take it for 9 days and then I don't take it again for a year, right? It seems like they're kind of acute moments. But we're kind of thinking about the wrong selective pressure there. So let me introduce another one. MIC is the minimum inhibitory concentration of antibiotics where susceptible bacteria are impacted. Essentially, that's where bacteria will die if they're not antibiotic resistant. And so traditionally, when we thought about the evolution of antibiotic resistance, we thought about things at that MIC threshold. So when you're given a course of antibiotics, you're told to take them all because it gets you over that threshold and makes sure it kills off all the bacteria. But what researchers are finding is that there's also this other condition, which is sub-MIC selection. This is the condition that's not enough to kill off susceptible bacteria, but the antibiotics are still present. They just aren't present enough to kill off the bacteria. And so you see this level of selection a lot of places. You see it in the organ of treated patients. So after I finish taking my run of antibiotics, I still have antibiotics in my body, right? And it's just slowly getting lower and lower in level. So it's no longer killing things, but it's still present. It's also in my blood if I took antibiotics. And here's the most important place it is. It is out in the world. Half of the antibiotics that we consume goes into the sewer through urine. And so remember, it's not just humans using antibiotics, but all these agricultural farms that are raising cows, chicken, pig, whatever, they're using antibiotics. And at least 10 tons of antibiotics are released into the environment a year via urine. So antibiotics are everywhere. It's in our soil, it's in our water, it's at low concentrations, but they're there. And that's really important. So the next slide, I'm going to give you a graph, and I want you to think through what that graph implies. So let me orient you here. On the x-axis is the concentration of antibiotics from lowest to highest. The green is basically no antibiotics. The red is the levels that you would have if you were taking antibiotics. And then we've got this sub-MIC area in yellow. The blue line is the susceptible strain, the strain with no antibiotic resistance, and the red line is the resistant strain. So using that, please go ahead and answer the question that pops up. So what I hope that you saw in this graph is that the benefit to antibiotic resistance is not just at the level of when we're actually actively taking antibiotics, but at this level, at the sub-MIC level, being antibiotic resistant, you still have higher growth, which means you can probably outcompete that's not antibiotic resistant strain, as long as there's a little antibiotics present in the environment. And unfortunately, there's often a little antibiotics present in the environment, all right? It's only when there's no antibiotics at all that a strain susceptible to antibiotics outcompetes the resistant strain. So that means selection is consistent in time. And that is a real problem. That means there's constant selection pressure to develop antibiotic resistance. Not because we're taking antibiotics correctly, but because we take so much antibiotics and we use so much antibiotics in agriculture that just out in the world, there are low levels of antibiotics that bacteria encounter all the time. So what do we do? What are the things that we can do to save antibiotics? Because without them, we're really going to be in trouble. So I want you to use your knowledge of evolution and suggest 2 to 3 strategies for reducing the spread of antibiotics. And you could think of this at a couple different levels. What could individuals in the population do? What could policymakers do? What could health professionals do? What could the agricultural industry do? And so on and so forth. And once you've come up with suggestions, I want you to write out an explanation for 2 of them about why these would work. And I want you to use the idea of fitness and things like that and migration, if you're using migration, to explain it. So use your evolutionary knowledge to explain it. And then I want you to rank them. What's going to be easiest to implement, what's going to be hardest, and explain why the top one would be the easiest to implement. And please, in this answer, include A, B, and C so I can see the different portions of your answer are present. All right, good luck. Okay, great. So I hope that you were able to come up with some stuff and that you were really thinking about how evolution can be helpful for advising people on what to do. And so now I'm going to go through a couple of suggestions that folks have made, organizations like the World Health Organization have made, to help prevent the loss of antibiotics. And maybe these are the same as some of the ones you came up with. The first suggestion is to reduce the use of antibiotics. So some possible policies that could help with that are doing things like avoiding antibiotic use when none is needed. So for example, when I go in with a bad cold, sometimes they give me antibiotics, but they don't actually test to see if I have a bacterial infection. That shouldn't happen. So when an infection is caused by a virus, we shouldn't assign antibiotics. We should test to make sure that there's a bacteria present, right, before we assign antibiotics. So another idea is we could discontinue the use of antibiotics in animal husbandry. So instead of getting that 11 million kilograms a year, we could just be putting into the environment what humans use. Another suggestion that was maybe at the level of the policymaker is avoiding economic situations in which prescriptions of antibiotics are profitable for the prescriber. So making it so that doctors or hospitals or pharmacists don't get cuts off of based on how much they prescribe. And doing some control and regulations of antibiotic marketing so that they're only actually used when a doctor thinks it's important, and prohibiting the sales of antibiotics to the public without prescription. Okay, so all of these policies would reduce the use of antibiotics, but I want to know what that would do. So go ahead and answer the question that pops up. Great. So what I hope you see is that this approach, this potential solution, all these policies are put into place to reduce natural selection. If antibiotics are used less, if they're less prevalent in the environment, then the strength of selection that favors the emergence and spread of antibiotic resistance also goes down. And so you're less likely, it's going to slow the spread of antibiotic resistance, and it's less likely to persist over time. So again, your knowledge of evolution is helping to give you strategies that we can use in the real world to protect something that's really important to us. Here's another set of strategies. These strategies all focus on increasing the use of various hygiene and infectious control measures. So some of these policies might be things like isolating patients with infections from other patients that don't have it. Improving hospital hygiene measures, right? So making sure that hands, instruments, the environment are all clean. We could improve animal hygiene and welfare on farms. Again, this would limit, right, the ability of the virus to spread. And we could apply best practice at all stages of production and processing of food, keeping it clean, keeping it at safe temperatures, using safe water on raw materials. Okay? So again, thinking about these policies, what mechanism of evolution are these policies addressing? So I hope you saw that what these policies are trying to do is prevent the spread of antibiotic resistance. So it may be in one patient, but if you isolate that patient from everybody else, those antibiotic resistant bacteria aren't going to spread to the next patient, right? It may be in one animal, but if you keep the environment hygienic, then it won't spread. Or if you butcher that animal and people are eating it, as long as you've processed it in a safe way, it won't spread, right? So all of these things are related to migration, and you're just reducing the ability of the antibiotic-resistant bacteria to spread. Okay, last approach. This approach is about how we develop new antibiotics. You may or may not have thought about this one, but here's the bad news. The pharmaceutical industry isn't really developing any new antibiotics right now. It just isn't profitable. It takes a long time. You have to go out, you have to isolate the protein, right, from soil bacteria. There's millions and millions of soil bacteria species, so it's just chance that you find the right one, right, that has one that's medically useful. And it's expensive, it takes a long time. They aren't doing it. But we could be doing it. And if we were doing it, there's some strategies that we could take among either choosing which antibiotics to develop or creating new ones, which could be the wave of the future. So if we were doing that, we could make sure to create antibiotics that is hard to acquire, right? So something about it is hard to transfer to individuals, or it's really hard for a mutation to occur there to make you resistant to it, right? So acquiring that resistance has a high fitness cost. And then also opportunities for other mutations to restore the fitness. So let's say you develop an antibiotic resistance, but it makes it so you can't process whatever food the bacteria eats. Well, that's a really high cost antibiotic resistance, and those individuals are going to die, right? But if some secondary mutation was able to help that bacteria recover their ability to feed and they had the resistance, then it can continue. But if you made it very hard for that second mutation to occur, then again, you would really limit the ability of bacteria to develop resistance. Okay? So once again, I want you to think about what mechanisms does this approach target? And it may be more than one in this case. All right, so what I hope you saw there is that this approach is targeting mutation and natural selection. So basically, you're trying to make it harder for a spontaneous mutation to make resistance, or if a spontaneous mutation occurred, harder for that mutation to exist in the population because it is so costly. Right? So basically, you're making it more costly to the bacteria's fitness. So mutation and natural selection are what you're using here. So again, these policies are all using our understanding of evolution in a real world context. So if we go back to our big question, or our big challenge of will antibiotics still exist in 10 years? Will they still have medical value, or will they no longer work? Right? We've talked about some policies, but there are two major challenges that we have to overcome. The first is that society has to resolve to use antibiotics in a more restrictive way, or it won't work, right? If antibiotics are just out in low levels in the environment, you're creating a consistent selection pressure that drives the evolution of antibiotic resistance. We can't have this if we want to still have functional antibiotics. This is going to require a global commitment to be effective, right? Countries are going to have to coordinate with each other. But if we fail to implement this in the next 10 years, antibiotic resistance will continue to evolve, and we will likely lose antibiotics. We also need the pharmaceutical industry to recommit to antibiotic discovery and development. If it's not profitable, we may need to create some new business models where maybe profit isn't the motive, or we somehow make it more profitable. And to do that, we need more scientists. We need more people with knowledge like yours of the structural biology, of bacteria, of chemistry, right? All these things that we have to understand to create antibiotics. So we need new models, and we need more people who are knowledgeable about biology. And that is a role you could play. And, I hope you saw this coming, we need to understand evolution. Understanding evolution will help us come up with the policies to prolong the activity of current antibiotics and help us think about ways to combat future antibiotic-resistant genes. Understanding evolution helps us come up with the solutions. Evolution is not just esoteric. It is something we are using actively to improve the lives of humans, to improve the lives of our community. All right? And it's not just here. Antibiotic resistance was one example. I think it's really important and not one that's talked about as much as it should be. And so I highlighted it here. But the last question I have for you is this. The world is facing many challenges. Where else do you see an understanding of evolution can help us make decisions or guide the future? We've talked about some of these in past units, but try to come up with 3 challenges and explain in 1 to 2 sentences how what we've learned about evolution could be used to address them. This could also potentially be the basis of your extra credit assignment if you are planning on doing that reflection. It doesn't have to be, but it could be. All right, so take a minute. Think about 3 challenges the world is facing that evolution could address. All right, y'all, with that, you have finished the final module. I've really enjoyed our class this semester. I know it was challenging as we switched online. I learned some new skills. I bet you all learned some new skills. And please, 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 if you have any questions as you're preparing for the exam, hop onto office hours. I'm going to send out an email with more office hours and links to the Zoom for those. And I hope that you all continue to stay safe and that your families and your friends stay healthy. All right? Have a great rest of the semester. I hope to see some of you on office hours. And for everyone else, congratulations on making it through. Bye.